Hello, this is Professor Swindell, beginning the podcast for the urinary system for the Anatomy and Physiology course, Bio 133. Before I get started, I just want to remind everybody this is Chapter 18 in our textbook, the Understanding Anatomy and Physiology. I also want to remind everybody to utilize the textbook resources that you can find online on Davis Plus. You require the code from the inside of your textbook. It will allow students to do practice of drop downs, labeling, definitions, and it's quite useful for your success. So let's talk about the urinary system. The structure of the urinary system, looking at this slide, you see most superiorly, you see the two kidneys. The two kidneys each have the adrenal gland sitting on top of it. We discussed the adrenal gland in more detail in the endocrine system. You see each kidney has a ureter leaving the kidney, which is bringing urine that was formed down to the storage area, which is the bladder. And we have one bladder and one urethra, which is the exit for urine. So the structures of the urinary system include the kidneys, the ureters, the bladder, and the urethra. The kidneys' location sits posteriorly retroperitoneal, meaning they're outside of the peritoneum, that serous membrane of the abdomen. They sit at about the 12th rib, underneath that floating rib there. So the ribs help protect, as does a layer of adipose tissue to protect that very important organ, as we'll see. It's about 4 inches long, 2 inches, 2 inches wide, and about an inch thick. And they extend from about T12 down to L3 of the vertebrae. As you can see in this picture, the right kidney is lower than the left due to the liver being on that side. This slide shows a cut of the kidney that allows us to see the different layers, starting with the outermost fibrous capsule that surrounds both kidneys. Other than protection, it doesn't serve another purpose, versus the renal cortex. We'll learn that that's where our nephrons are found. So the cortex is outermost. Cortex, you remember hearing about cerebral cortex, cerebellar cortex, adrenal cortex, so it's the outermost. Medulla, middle, I usually say, or inner in this case, where we have as a result of some cortical tissue extending into the medulla, there are pyramids, renal pyramids that are present, and we'll learn that that's where we have the collecting ducts for urine. From the ends of the renal pyramids, we have renal papilla, which from there we have the calyces, which begin like the funnel that's collecting the urine. So we have a minor calyx, which is singular and covers one papilla. And then we have a major calyx, which is where more than one minor calyx has drained urine. And the pelvis, which leaves the kidney as the ureter. So it looks like a enlarging funnel. And the ureter is how urine gets to the bladder. You notice in this picture we have an area of the kidney called the hilum, which is how blood enters and exits the kidney through the renal artery and renal vein, and how the ureter leaves, but also we have nerves that enter in through that hilum as well. This question, blood vessels, nerves, and ureter enter and leave the kidney from we just discussed it, so that's the hilum. The others we know are incorrect. The renal pelvis is before the ureter. The calyx is where urine is collected at the end of the medulla or the renal pyramid. and the renal pyramid's part of the medulla. There we go, we were right. So let's talk about renal circulation. We mentioned that the renal artery brings blood, oxygenated blood, to the kidney. And think about it, the renal artery comes off the abdominal aorta, bringing oxygenated blood from the left side of the heart. Just like we've discussed in every other system thus far, there's branching where it gets smaller and smaller and smaller arteries that end up getting super small, going up to the renal cortex. And in reverse, venules or small veins get bigger, 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 leave the kidney through the renal vein, and return to general circulation through the inferior vena cava back to the heart. In the cortical area, we have the nephrons, which are the units of the kidney that do filtration, and they generally are out in that cortex region. As we'll see in the next slide, the loops of Henle, or the nephron loops, actually can extend into the medulla, but most of the nephron is in the cortical area. As far as renal circulation goes, you should know that from the renal artery, smaller, 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 to arterioles, we have the afferent arteriole that brings blood to the glomerulus. The glomerulus is just a bundle of capillaries. And from the glomerulus, we have efferent arteriole. So anything that doesn't pass through that glomerulus, and it'll make more sense in a minute, anything that doesn't pass through the glomerulus will stay in the efferent arteriole. And the efferent arteriole actually becomes the peritubular capillaries. So in the nephron, there's actually two capillary areas, one the glomerulus, and two peritubular. And as we know, what happens in capillaries is waste products go one way and nutrients go the other way. In the case of the kidney, it's happening in two places. We have the opportunity for things to go back and forth between the nephron tube as well as the peritubular capillaries. Pretty interesting. So from the peritubular capillaries, we have the venules, which get bigger, 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 and then leave the kidney through the renal vein. And that's shown on this picture. So this is a picture of a nephron, and you see in red and blue the blood supply, and in the beige color tube is the renal tubule, or tubule will be, it's either just plain old tubule, but it is between the blood and the tube that ultimately we have urine formed, and we'll talk about the processes of urine formation in a minute. So we talked in the nervous system how the brain is obviously a very important structure where it receives a significant percentage of the blood that's circulating in the body from the heart. And the same thing is true with the kidney. So 20% of the blood that's pumped with the heart with each beat, each minute, goes to the kidneys. And the job of the kidneys is to filter blood, make urine, filter blood, get rid of the waste products, keep what you need to keep, and get rid of the waste products. So it's very important, and you could tell the importance by how much blood it's getting. That's a trick. So again, staying with this picture, you have the afferent arteriole to the glomerulus, to the efferent arteriole, to the peritubular capillaries, and venules to vein and back out the renal vein. That's essentially the blood flow that you need to know, and that the nephron is the area of the kidney where all the good stuff's happening, all the filtration is occurring. So the nephron is broken up into two parts. One, the renal corpuscle, which is made up of the Bowman's capsule or glomerular capsule, both the same thing, and the glomerulus. So we have the glomerulus, which is that bundle of capillaries in the center of the slide, and Bowman's capsule, which is a double membraned structure, and we'll see how it works. The combination of that capsule and the bundle of capillaries. What happens as blood is coming in through the afferent arteriole, the pressure in there is greater and pushes things through the walls of the capillary directly into the capsule. And if you look at the capsule, that's continuous with the tubule. So blood comes through the afferent to the glomerulus. Pressure pushes things through into the capsule, into the tubule. So the glomerular capsule, or Bowman's capsule, surrounds that glomerulus. Your book describes it interestingly as, imagine your fist pushing into a inflated balloon. If your fist is the glomerulus and the balloon is the Bowman's capsule, you see how the capsule kind of surrounds the glomerulus. So what gets pushed through the capillary would be fluid and some solutes that are in there, and we'll talk a little bit more detail. So renal corpuscle, capsule, and glomerulus, and we'll see the second part of the nephron in a second. Here we go. The renal tubule is the second part. So just like you needed to know the blood flow through the kidney, you need to know the filtrate flow through the nephron. So we had, for blood flow, we had afferent to glomerulus to efferent to peritubular, capillaries to venules, veins, and out. In the case of the tubule, we have the proximal convoluted tubule, which is closest to the glomerulus, the loop of Henle, which has two parts, the descending limb, which is first, and then the ascending limb. And then we have the distal convoluted tubule, which connects along with distal convoluted tubules from other nephrons to the collecting duct. And at the collecting duct is where you're going to see urine. So the flow of filtrate, anything that left that glomerulus and went into the Bowman's capsule, is now called filtrate. And the first section it comes in contact with would be the proximal convoluted tubule. So when you think of a nephron, it's not as clear as this picture, in that this picture is really just stretched out. It's actually more compressed and more challenging to evaluate. But that proximal convoluted tubule right off of that Bowman's capsule contains microvilli. And if you remember back to the chapter 3 or so when we talked about cells, the purpose of microvilli is to increase surface area for absorption. So those microvilli in that proximal convoluted tubule increase the amount of substances that can get absorbed through it. So remember, we also have peritubular capillaries. So absorption means substances are going from the tube and then going to the bloodstream. So after the proximal convoluted tubule, you have the ascending descending and then ascending limb of the loop of Henle. Sometimes they'll call it the nephron loop. And the distal convoluted tubule, then the collecting ducts. So the filtration units of the kidney are the nephrons. The renal corpuscle is a part of the nephron. The glomerulus is inside the corpuscle, or part of the corpuscle, and the pelvis is how urine leaves the kidney. It connects with the ureter. So the answer for this question is C, the nephron. Yay, us. So let's talk about urine formation. Urine formation requires 3 processes. Glomerular filtration. Remember, filtration as a transport method is just based on pressure. Tubular reabsorption, which is from the tube to the bloodstream, and tubular secretion, which is from the bloodstream back to the tube. So 3 processes are involved in urine formation, and it's those 3 processes, how much gets reabsorbed and secreted, as well as how much got pushed through, that determines what our urine's going to look like. So let's start with glomerular filtration. So this slide shows the corpuscle. It shows the afferent bringing blood into the glomerulus, efferent leaving the glomerulus, and you see that the Bowman's capsule surrounds the glomerulus. So because of that pressure, you see in the bubble picture, you see that things get pushed from the glomerulus through pores in the capillaries into the capsule. What is obvious in this picture is that red blood cells and plasma proteins are bigger molecules, and they don't pass through the pores. Just like my coffee grounds don't go through my coffee filter, the red blood cells and proteins don't go through the pores. Other things that are dissolved in blood will go through, such as different electrolytes, glucose, amino acids, or broken down proteins, vitamins, waste products, and they go directly into the Bowman's capsule. One thing that's interesting is if somebody had red blood cells or a protein in their urine, you know that there's a problem with the filter. Just like when I get coffee grounds in my coffee, that means there's a problem with my filter. So it's telling us when we do urine assessments, we've got a problem, and it could be at this filter area. So the glomerulus filters approximately 180 liters of blood every day. 60 times our normal blood volume. But as we know, we only void 1 to 2 liters of urine. So most of what gets filtered gets reabsorbed. What's happening as blood is being filtered, it gets concentrated. The urine gets concentrated as it's going through. Everything else goes back into the bloodstream. Pretty cool. So glomerular filtration rate equals it's the amount of fluid that's filtered by the kidneys, and it's about 180 liters each day. As you know, we talk about normal, so that when we hear abnormal, it makes more sense. When somebody has kidney failure, whether it's acute or chronic, you'll see the filtration rate decrease. So instead of 180 liters per day, they're processing significantly less. So if it's not getting rid of waste products, that means waste products are in the bloodstream. So that presents its own problems. So we evaluate glomerular filtration rate. Think about med excretion. If somebody's not filtering blood and getting rid of waste products, then the person could be at risk for toxicity. So the regulation of glomerular filtration rate is by our body is a constant thing, in that despite changes in blood pressure, glomerular filtration rate should be the same throughout. If the flow is too high, what would happen would be that there'd be too much lost, too much water, too many nutrients. If it's too low, then the tubules may reabsorb toxins back from the tubes into the bloodstream, and we don't want that either. So there's a couple of processes that will regulate glomerular filtration rate. Before I continue, I just want to mention the life lesson on the bottom of 361, talking about the effects of hypertension on kidney damage. So in addition to diabetes, hypertension can wreak havoc on kidney function. As blood pressure goes up, our systemic hypertension goes up, what happens is the glomerular pressure goes even higher. And as a result, there's damage to those capillaries. They could actually burst, and as a result of that, become scarred. And as we've talked a number of times, whenever there's scarring, we have decreased function. In addition to the damage to the capillaries, we also have the problem with hypertension causing or contributing to atherosclerosis, where there's lying down of plaque and buildup of plaque in the blood vessels. So that could be the renal arteries, where there's decreased blood flow to the kidneys, and therefore can contribute to further kidney damage. So let's see how our body controls flow. And one of them is the renin-angiotensin-aldosterone system, or RAA. Sometimes you'll see RAAS. So that's a key way of maintaining blood pressure and therefore maintaining a steady GFR. So how it works is, when there's a decrease in blood pressure, there's an area around the afferent arterioles that will respond to that decrease in blood pressure, decrease in blood flow to the kidneys, by releasing the enzyme that's called renin. And renin will convert the plasma protein that's just floating around in the blood, called angiotensinogen. It'll convert it into angiotensin 1. Angiotensin 1 travels around in the bloodstream, goes to the lungs, and in the lungs there's an angiotensin converting enzyme, otherwise known as ACE, that converts angiotensin 1 into angiotensin 2. Angiotensin 2, one of the things that it does is it stimulates the adrenal glands to produce aldosterone. And aldosterone, remember, is one of the corticosteroids, salt, where it causes the kidneys to hold on to sodium, therefore hold on to water, therefore increasing blood pressure by increasing blood volume. So in pharm, we talked about RAA and some meds used to lower blood pressure, but one of the other things that RAA does, or angiotensin 2 does, is cause vasoconstriction. And vasoconstriction, we know, contributes to hypertension. So the meds, ACE inhibitors, block the enzyme that converts 1 into 2, and ARBs are angiotensin 2 receptor blockers, will block the receptor for angiotensin 2, so therefore no vasoconstriction, therefore no aldosterone. Pretty interesting. But RAA is our body's way of regulating blood pressure, therefore regulating essentially glomerular filtration rate. An interesting fast fact on the bottom of 362 includes that during shock, when there's, like, say, hypovolemia, you have the sympathetic nervous system kick in, causing vasoconstriction of the afferent arterioles, which causes blood to be diverted from the kidneys to keep it in the brain and bloodstream, or the heart and the brain. And so the glomerular filtration rate, because that constriction of the afferent arterioles trickles down to almost zero. So interesting how our body opens up flow and decreases flow. So let's look at the next slide. In this slide, we're talking about tubular reabsorption and secretion. So remember, the three processes of urine formation is filtration that occurs in the glomerulus. And then we have reabsorption and secretion. Reabsorption is from the tube back to the bloodstream, and secretion is from the bloodstream back to the tube. So this picture is all stretched out, and it doesn't really show the capillaries surrounding the tube, but imagine it's wrapped around that tube pretty closely and allows for things to go back and forth. So as soon as substances come into the glomerular capsule and start going into the proximal convoluted tubule, which is represented on this slide in green, you see so much stuff automatically going back right away back into the bloodstream. So it got pushed through and it goes back to the bloodstream. And then in the rest of the parts of the renal tubule, you'll see things going back and forth. In the descending loop of Henle, you see that there's water leaves the tube and goes back to the bloodstream. And on the ascending, which is in pink, you see sodium leaving the tube, but you don't see water. Normally you see sodium, therefore water. And one of the issues is in this area, the wall is thicker and water doesn't move through there so well. And then in the distal convoluted tubule, you see some sodium, water, potassium going from the tube back to the bloodstream. You see some things from the bloodstream back to the tube, such as hydrogen ions. And then you see in the collecting ducts, you have a last ditch where you can have water reabsorb back to the bloodstream and further concentrating that filtrate that's in the tube and resulting in the urine that we know. So average urine output is anywhere from just under 1,000 to 2,500 mLs, and is influenced by how much we take in. So drinking a lot, we're going to have more dilute urine and increased volume, and a fluid restriction is going to cause a decreased urine output. Some meds can influence, like a diuretic is going to cause an increase in urine output. Sodium in the excess sodium in the diet is going to cause water to be retained, because where there's sodium, there's water. That's why after you eat a salty meal, you'll be thirsty, because it wants to even out that concentration. So what I want to point out is two things. One, it's abnormal to have glucose in our urine. It shouldn't be there normally. So if you look back at the beginning at the glomerulus, you see glucose gets pushed from the blood into the capsule, therefore into the proximal convoluted tubule, but immediately almost all glucose goes back into the bloodstream. Pretty amazing. Because remember, our body needs glucose for what? I'm not even going to answer because I know you know the answer. Our body needs glucose for blank. I'm not going to tell you. So you have that glucose get taken back into the bloodstream almost immediately. So most sodium is reabsorbed from the glomerular filtrate right away in that proximal convoluted tubule, as are lots of things like glucose, like water, etc. And that's the area. The distal convoluted tubule has some reabsorption of waters and salts and potassium. The collecting duct, essentially you see water, and the glomerulus is where that filtration occurs, where things leave the blood and go into the capsule. So the best answer would be proximal convoluted tubule. Let's see. That is correct. We're batting 100. All right, let's take a look at hormones and the effect that they have on the urinary system. Some of this may be repetitious because we talked about some of these in our cardiac lecture. But before we do, if you look back at page 363, just to stress that in the descending loop of Henle is where you see the water leaving the tube going into the blood, and then in the ascending is where you see sodium and chloride leaving from the ascending loop limb into the capillaries, where water isn't on that side. Water's on the descending side. That's an important difference to note once again. So the hormones that affect the urinary system, aldosterone comes from the adrenal cortex, causes the kidneys to reabsorb sodium and therefore water, and therefore increase blood volume and blood pressure. And there's an excretion under the influence of this hormone of potassium. Atrial natriuretic peptide, it's known as ANP, causes the kidney to excrete sodium and water. So this is released from the muscles of the heart when there's an increase in blood volume. So the atria is stretched, and therefore this hormone or peptide is released, tells the kidney to get rid of salt, therefore water, therefore decreasing blood volume, blood pressure. Antidiuretic hormone is released or made in the hypothalamus, stored in the posterior pituitary, and is released to prevent dehydration. Diuretic would be a medication that causes our kidney to lose urine, increase volume of urine, lowering blood pressure. But antidiuretic would be to not lose urine. So what's going on is water is leaving the tube, going back to the bloodstream. As a result, increased blood volume, increased blood pressure. And last, another hormone that affects the urinary system is parathyroid hormone. So parathyroid hormone, as we talked about in endocrine system, that T looks like an arrow going up to me, and what's going on is the kidneys are reabsorbing calcium. Calcium is being reabsorbed because blood calcium level is low. So parathyroid hormone is going to increase blood calcium level. And there's an excretion of phosphates. Calcium up, phosphate down. Phosphate up, calcium down. They work opposite each other. So let's take a look at what urine looks like. Like a lot of the fluids in our body, we have most of the fluid is water. And then we have a portion of it that's dissolved substances. On page 365 shows what a urinalysis will show us. For example, color. We're looking at color where urine is generally a pale yellow to an amber color. And odor should be mild. A pungent smell might be stale urine that as a result of bacteria converting the urea into ammonia. That's what gives it that strong ammonia smell. Fruity smell could be from diabetes, that there is glucose in the urine. And a rotten odor, a foul odor, could be that there is a urinary tract infection. Back up to color, when somebody's urine is very light, That could be that they are potentially overhydrated. You'd have to be concerned about volume as well. But darker urine is usually not enough hydration. Cloudy urine could be telling us that there's organisms in there, bacteria, so an infection. So we look at the color of the urine, it tells us a story. Remember that some meds can change the color of urine as well. I believe we talked about rifampin and one of the tuberculosis medications. So we look at color, we look at or smell odor, and we also measure specific gravity, which tells us how much solids are in the liquid. So a more concentrated urine is going to have a higher specific gravity, and a lower specific gravity is going to be more fluid than solids. That kind of gives us an indication of how hydrated they are. And last would be pH. Generally, a pH around 6 is considered normal. So a higher pH is telling us it's more base or alkalytic, and a lower pH would be acidotic. So in addition to these things, we'll also look at blood to see if it contains look at urine to see if it contains blood, because we talked about that blood in the urine is not normal. That's telling us potentially that there's a problem with the filter. There shouldn't be glucose. We're looking at albumin or proteins, as well as ketones or bile pigments. These are things that are normally not in urine. The presence of them in urine is telling us that we may have a problem somewhere else in the body. So important to examine urine, just like we look at skin and nails and hair and eyes that tell us a lot of things about a person's health, so does urine. You don't even have to touch somebody and you can come up with a lot of things. Before we continue, the other issue with urine, we want to assess volume. So we normally between 1 liter to 2 liters of urine output each day. When we have less than 400, we're quite concerned. It's called oliguria, because that's not enough urine to clear the waste products from our body. In some disorders, such as diabetes, we might see polyuria, increased urine output. Diabetes insipidus is a disorder where there is too little ADH. Remember, ADH tells our kidney hold on to water. Well, if there's little or no ADH, the body's going to be losing a lot of water. And that could be a problem. That's diabetes insipidus. We also need to talk about creatinine. So creatinine is something that should be steady eddy. You see the level of creatinine in the blood and compare it to the urine, because the kidney should be getting rid of the same amount of creatinine fairly consistently, and the blood level of creatinine should be fairly steady. When you see creatinine levels going up in the blood, that's telling you that the kidneys are not able to get rid of it. And that is one of the indications that the kidneys are not functioning well. Right, let's take a look at how urine is stored. So the kidney makes the urine by processes of filtering, reabsorption, and secretion, and it's collected in the pelvis to the ureter and goes down into the bladder, where it's stored. Generally, it's stored a few hundred mLs, and we get the sense that there's urine and we need to go. But because of some sphincters, we can actually hold our urine, and like nurses often do, you go at the end of your shift. But that's not good. You should be emptying your bladder more often. But the bladders will store the urine until it can be eliminated. So from the kidney, we have the ureters, and the uriners from each bladder from each kidney enter into the bladder. The next slide, we're going to see a close-up of what the bladder looks like. But before we do, let's talk about the ureter. It's a fairly small tube. It's long in length. It's about 10 inches in length, but it's narrow in diameter. So if somebody were to have kidney stones where there's crystallized, it could be calcium or uric acid or proteins or infectious material in the kidney, it could actually become lodged in the ureter and block urine flow. So urine's trying to leave the kidney, go down the ureter where it can't continue, so it backs up and can cause a condition that's called hydronephrosis, where fluid kind of fills the areas of the kidney and can cause kidney failure. So it's a keeping urine flow is important, and we'll talk about how urine leaves the bladder in a minute. So looking at the urinary bladder, you see the detrusor muscle, which is the smooth muscle that surrounds the bladder. It's got 3 layers. It seems like we have a lot of that. We saw 3 layers of muscle, or we will see 3 layers of muscle in the stomach, but sometimes we have a longitudinal layer and a circular layer through the GI system. In this case, we have 3 layers of smooth muscle in our urinary bladder. The bladder, as we know, sits about at the symphysis pubis. It's generally not palpable when it's normal. When somebody is retaining fluid, we might see it above the symphysis pubis or the pubic bone. So you can see that the ureters enter into the bladder actually low. You see it in the bottom third of the bladder. You think that it enters into the top and then urine drips down, but it actually enters in fairly low. The rugae are the folds that allow for expansion of the bladder, and we'll see that similarity in the stomach. There's rugae in the stomach that will expand, allow for expansion. So normally it's a small hollow muscular cavity, and then when urine fills it, it can stretch. And we talked about the type of tissue there. It's that transitional epithelium that can change shape. At the bottom of the urinary bladder is a trigony, where it's just a triangular, triangular area at the bottom of the floor of the bladder. The next thing I want you to note is both the internal urethral sphincter and the external urinary sphincter, where the internal urinary sphincter is under involuntary control. It is what will hold urine in the bladder, whereas the external urinary sphincter is voluntary. It's actually skeletal muscle versus smooth muscle, and we have control over that that we gain through our toilet training as toddlers. So we can hold our urine until we have the right time or the right place to void. The internal sphincter might be relaxed when there's a certain volume in there, but we can hold it by using the external urinary sphincter, that skeletal muscle there. The external urinary meatus is the opening of the urethra that leads to the outside of the body. This is where the insertion of a catheter would occur, the urinary meatus. The urethra, this is a good picture showing the difference in the size of the urethras between a male and a female. The female's is only about an inch or so in length, and it exits in front of or anterior to the vagina. So when you'll often hear people, nurses, talking about inserting a urinary catheter, that their first attempt is in the vagina, so they'll leave the catheter there, find the urethra, which is going to be superior to that if the person's lying supine, anterior to that if the person's lying supine, and allows you to find that urethra better. And in the male, you see that the length is closer to 8 inches. One thing I want to point out is close right underneath the bladder where the urethra exits is the prostate gland. And as men get older, there could be some hypertrophy of that gland that causes a difficulty in urination, emptying the bladder, starting the stream, and that gets enlarged. But also can present a problem when you're inserting a catheter. I don't know if you talked about that in 105, but that prostate gland right before it gets to the bladder can provide some resistance. So the difference in male and female, another difference between male and female besides the length of the urethra, is the fact that in men, the urethra is the exit for urine as well as semen, so both reproductive and urinary, where that's not the case in a female. You have the bladder and the vagina. So let's talk about how urination occurs. So a couple things. Micturition is another word for urination, as is voiding. You'll hear that. And what happens is, this is a great description on page 368, when the bladder contains about 200 to 250 mLs of urine, the bladder stretches and there's receptors, nervous system receptors in the bladder that will send impulses to the lower part of the spinal cord, the sacral region. The spinal cord then sends information back out to the bladder wall to contract and for the internal sphincter to relax. And when that happens, we'll have voiding occurs, involuntary, unless the brain overrides it. So why would the brain override it? Would be, we're not in the right place to be urinating. So the brain has the ability to override the impulse to void because the stretch receptors, in addition to sending information to the spinal cord, also send information up to the pons. So pons, if you remember back in the nervous system, said it's involvement in respirations, but the other one is urination. So what the pons does for urination is it takes the information from the stretch receptors, you gotta go, and sends information or has information from other parts of the brain, like the cerebrum, that says, you know what, I don't think it's the right time to urinate, nor am I in the right place to urinate. So therefore, as a result, the urinary sphincter is contracted. External urinary sphincter is contracted. So if it's time to urinate, is appropriate place, the brain sends signals to the bladder wall to contract and the external sphincter to relax, and then you could void. So pretty interesting, and it's that control we gain as toddlers of our ability to hold on to that external sphincter and not urinate. So the other thing, that the last concept is about renal insufficiency and dialysis. So we could have insufficiency or renal failure that happens suddenly, more acutely, or it could happen chronically as a result of hypertension and diabetes and such. But ultimately, it's failure of the kidney to be able to do what it does. If it's acute, it potentially can be reversed. Chronic is progressive over time, can get worse. So the treatment includes dialysis, which can be done either using the body as the filter, or like as in peritoneal dialysis, or hemodialysis, where somebody has to get hooked up to a machine that filters the blood. We'll talk about that in more detail, but somebody that has really poor kidney function is unable to filter blood and therefore unable to get rid of waste products. If somebody has kidney failure and does not get dialyzed, they potentially could die because of the buildup of waste products in the bloodstream. So it's an important concept to remember. For some people, they're receiving dialysis because they're waiting for a transplant. Some people receive dialysis and never get a transplant. They may suffer from complications. So I hate to end on this sad note, but let's take a look at the last question. This question is: a triangular shaped smooth area on the floor of the bladder is called and the answer would be D. The rugae are the folds. The papilla are the ends of the renal pyramids. The cortex is that outermost region of the kidney that contains the nephrons mostly. So let's see what we got. Yes, we are correct. The answer is D. So because we ended on a good note, this is the end of the podcast. Remember to get online, do some textbook resources, read the book, answer the questions at the end of the chapter in preparation for any examination. Alrighty, this is the end of the podcast.